You know it well, the RBC looks like this, isn't it? What is the shape? Very good. We have discussed it in our earlier class. What is the size of this? Diameter? 7.2 micron. And what is its thickness at the center? 1 or less than 1 micron. Okay, and at the periphery, what is the thickness? 2.5. Excellent. And the center of these, when you see under the microscope, the center of the cell will look pale, isn't it? Whitish. Why? Their hemoglobin is less. It is having less color at this part since it is very thin. So that's why it appears white. Okay. So the RBCs look under low power of microscope like this, and these are the shapes we know it well. The center part is pale, and these things we already size, this much color is red due to hemoglobin, and the normal count you have to remember all these values. Okay, you know it well. We have discussed in our earlier class. So this may be asked in biopsy exam, so you must know the normal values. And its surface area of RBC is 140 micrometer square, and its volume is 87 micrometer cube, and it ranges from 78 to 94. We will be studying, not in this class, in a separate class, different hematological indices. With these indices, we will predict what is the size of a RBC in your blood. Okay, how much hemoglobin it contains, and what is the concentration of hemoglobin. So different like MCV, MCAs, MCAC, color index, this sort of different indices will be knowing, and these will be having clinical relevance, clinical importance. And the shape is inside, it is biconcave. And what is the advantage of this? These are the advantages. One is to increase the surface area. Why to increase the surface area? Its main function is diffusion of its exchange of gases, carbon dioxide, oxygen with the tissues. Okay, in the tissues and in the lung. So for this area, higher the surface area, higher will be the diffusion membrane, so higher will be the exchange. That's why it is very important to increase the surface area. And another thing, when an RBC, you have studied it, is placed in an hypotonic solution, in isotonic solution, and in hypertonic solution, what happens to the RBC? In hypotonic solution, RBC swells. So, it can swell. Why? Because it is having a lot of surface. Okay, it can accommodate fluid within this. So, up to a certain limit, and because of this, it will take the shape of round shape. So, this type of RBCs are called spherocyte. It takes the shape of sphere. So, that's why it is called spherocyte. Normally, if it is found in our blood, that is abnormal. It is a pathological condition called hereditary spherocytosis. Here, these spherocytes are very fragile. Whenever they try to seize, when they become oval, this type of takes sphere shape, they cannot easily pass through the capillaries because capillary diameter is less. So, and their outer membrane is fragile. So, what will be there? What will be happen to the RBC? It will break down. Okay, and this is called hemolysis. Hemoglobin will be coming out of the cell. Now, I will discuss what will happen to this type of, after hemolysis, what happens. Okay, so that is, moreover, because of its shape, it can easily pass through the capillaries. These are the advantages. Now, what is there by which it can perform these duties. So in the cell membrane, below the cell, within the cytoplasm, there are cytoskeletons. What? Cytoskeleton. You have got skeleton. Because of this skeleton, you are having a particular shape. You are not like jellyfish. They are not having. So you got a shape because of these bones. Similarly, the RBC is also having skeleton. These are called cytoskeleton. What are these cytoskeleton? You will try to remember this. Okay, one is actin. Okay, I am coming to this. Okay, So these are the cytoskeletons. This is the membrane. And these are the cytoskeleton. The main cytoskeletons are, one is actin, another is spectrin. This spectrin is protein in nature. Since it is protein in nature, all proteins are synthesized in polyribosomes of the cell. How do they synthesize it? They get the information from the nucleus. What is there in the nucleus? Chromosomes. So in the chromosomes, the DNAs are there. So genetic codon is there. So they produce one mRNA specific for this protein. It is a messenger. That's why it is called messenger RNA. Now this messenger RNA from nucleus will travel to through the endoplasmic reticulum, and there are ribosomes. Now this thread, mRNA thread, will pass through the ribosomes. Now the ribosome acts like tape of a tape recorder. Have you seen tape recorder? Have you seen the tape? So it's like that. It will be passing through this. When this will be passing through this ribosome, ribosome decodes this message. Okay, first is lysine. Okay, in the cytoplasm, a lot of amino acids are there. So, who knows lysine? Oh, you know lysine. Transfer RNA. You go and catch this lysine. Okay, so it will be brought to the ER, endoplasmic reticulum. Line it. So, again, after this, glutamic acid. Okay, get the glutamic acid. So, lysine, then glutamic acid, then like this, one after another, amino acid will be joined together through peptide bonds. And a peptide chain will be formed. A protein will be formed. And now they will be packaged. Where? In the Golgi bodies. And when if it is needed by the other cells, then it will be exocytosed into the extracellular fluid. From there, it will enter into the blood, it will be circulated throughout the body. Like this, proteins are also hormones that are protein in nature, they are also synthesized like this. Okay, so this spectrin, actually, what is information is where? In the genetic code. Now, if this genetic code is disturbed, mutated, what will happen? Spectrin will not be formed. So there will be absence of spectrin, and it will cause what? The shape, RBC will be unable to maintain its shape. As a result, what will happen? It will get the structure. There is no spoke, like an umbrella. Okay, so nobody, or there are stings. No, you can think it as stings. No stings, so it will be taking shape of a spherocyte. So this condition is called spherocytosis. As I told, these are fragile. These are very prone to hemolysis. So they should undergo hemolysis. The person, if it is hemolyzed, hemoglobin is degraded. What will happen to you? What will happen to the patient? Who will carry the oxygen to the cell? None. So what will happen to the cell? Without oxygen, can they produce energy? So what will happen to the cell? So ultimately, the patient may die. So this is the consequences of spherocytosis. You know it well? What is this? RBC? This RBC is less, no? In which condition it is in? Anemia. Good. Here it is much more. In which condition it is seen? Polycythemia. There is an abnormal polycythemia. If I take you to a high altitude, what will happen? You will feel hypoxia, and this will cause liberation of erythropoietin from your kidney. Okay, and this erythropoietin will stimulate my bone marrow, and stimulate bone marrow for increased production of RBC, means increased production of hemoglobin, means more oxygen will be now able, the person will be able to carry more oxygen to the cell. Now the person will not be feeling hypoxia. Okay, so polycythemia. So it is a normal condition. It is a normal condition. It's not a pathological condition. But in case of polycythemia vera, these RBCs cannot function well. The lot of RBCs are produced from the bone marrow under abnormal condition, and this condition is a cancerous condition, and it is called polycythemia vera. Okay, VERA. So this is seen in polycythemia, or in case of polycythemia it will be much more. So polycythemia vera. Now, the normal blood contains 13 to 15 gram of hemoglobin per 100 ml of blood. How much? 13 to 14. Okay. And one RBC contains 250 to 270 millions of hemoglobin molecule. Lot of hemoglobin molecule. And there, what is the weight of this hemoglobin? It varies 27 to 33 picogram. Okay, you can assess it in the laboratory by measuring MCS, mean corpuscular hemoglobin. I told you there are the hematological indices. When we measure this hematological index and find a value within this range, then there is no anemia. Okay, when it is more than this, then in macrocyte, cell is larger in size, obviously, macrocytic anemia. When it is less than 20 picogram, it is hypochromic macrocytic anemia, SOTA. Okay, and its molecular hemoglobin, remember this point, every molecule of hemoglobin will contain how many? Heme? 4. How many? 4. And every molecule has hemoglobin, and globin is the protein part. And how many globin chains are there? 4. Okay, for each heme moiety, there should be 1 globin. So all of you are having 4 globin in each molecule. And out of these 4, 2 are alpha and 2 are beta. 2 alpha chains, 2 beta chains. I will be describing the structures soon. Each molecule of hemoglobin carries every heme part. Every heme, it is the heme part that carry oxygen. So one molecule having 4 heme, so it is capable of carrying how many oxygen molecule? 4. Okay. So remember this point also. Each heme moiety can bind and carry 1 molecule of oxygen, as I told. And another important thing, now you can calculate. 1 gram of your hemoglobin, 1 gram of 1 gram of hemoglobin can carry, its main function is to carry oxygen. How much? 1.34 milliliter. Okay? Now you can calculate. If I take 100 ml of your arterial blood, how much oxygen it will be carrying? Very simple. Your arterial blood is having, suppose, 15 gram of hemoglobin. It is the normal percent of hemoglobin. 15 gram percent, multiply it by 1.34. How much it comes? It comes, why you are late? Next day it should not happen. And what would be the value? 1.34 into 20? Near about 20 ml. 20 to 20. Now you can predict how much, if your hemoglobin is less, your arterial blood will be carrying less oxygen. If you are anemic, less oxygen will be supplied to the tissue. You will be what? You will feel hypoxic, you will be less energetic, you will be lethargic. No, I will not go to college, I am not feeling well. So you will be fatigued, less energy, it is there. From here to here, I am not able to go there. So these will be the conditions, okay. Now the functions of the RBC. Okay, everybody know. The RBC is having hemoglobin and hemoglobin carries oxygen. So our function of RBC is carrying oxygen from lung to the tissues. Okay, how it carries? As I said, the hemoglobin of it is having iron. This iron can bind with oxygen. Okay, with this, and what is the compound? Oxyhemoglobin. It can also transport carbon dioxide. How? Not by the iron, but by the globin chains. In the globin chains, there are amino acids, and one of the amino acids is having free amino group, and this amino group can combine with carbon dioxide to form. Is that clear to you? Every amino acids, every chain is having a terminal amino acid. One terminal is having free carboxylic group, another terminal is free amino group. So that amino group can combine with, form a compound, loose compound. This is called carbamino compound. So in this form, they can carry carbon dioxide also. And one more important function is hemoglobin can act as buffer. We said in plasma protein, they can act as buffer, because they can be ionized. So here we are having globin, and they are having amino acids. They are ampholytes. They are having positively charged NH3 plus at one end, another Cu negative. Okay, so whenever hydrogen ion is added, this hydrogen ion will be combining with the Cu negative carboxylic group. So neutralized. If I need, under the basic condition, basic condition means there is less hydrogen ion. So solution needs hydrogen ion. But how it will be coming? NH4, it will release one of the hydrogen ion to the solution, and the pH will be maintained. Okay, and this will become into NH2. Okay. So in this, is that clear? So it takes a major part. This hemoglobin takes, because see, plasma protein, how much it is? Per 100 ml? 6 to 8 gram. And where is hemoglobin? Much more. Almost more than double. So its buffering action is much more than the plasma protein. Can you remember this? Okay. So try to remember this point. Okay. Next. What is your blood group? Blood group? Yes. His blood group is AB positive. And what is your blood group? B positive. Who gave this identity? Who gave this identity? Which cells of your blood gave this? It is the RBC. It is the RBC gave you this identity. Okay. Why? How it gave? Because the RBC cell membrane contains antigens. These antigens are of two types. One is A, another is B. If you, who is AB? You. He is having both the antigens. That's why his blood group is AB. And you are B positive. B positive. Forget about positive. So you are B. Okay. So your blood group is, your antigen is B on the RBC membrane. And anybody O group? So what happens to you? No antigen is present. Either A or B, both are absent. And A, blood group A, having only A antigens. Okay, so this identity And this is it prevents mismatch when you need blood. So, depending on whether my blood matches with you, it is determined by these antigens. So these are very important, isn't it? And I told you to forget about positive or negative. So there is another antigens, and this antigen is called RS antigen. So RBC surface also contain this antigen. So if it is present, RS group, and this is blood group and blood type, whether you are having RS or not. RS antigen or say blood grouping and blood typing, 2 words. So whenever you will be writing your group, so write group A, type RS positive or negative. Clear? Okay. And it is the hemoglobin. It is the hemoglobin that gives red color to the cell, but other cells are not colored. Okay? And another thing, RBC is having one receptor for nitric oxide, for NO, nitric oxide. Nitric oxide is physiologically important. It is a vasodilator. Blood vessels are dilated by nitric oxide. So unnecessary nitric oxide, since they are having, say, if there is excess production of nitric oxide in the blood, so what will happen? This excess nitric oxide binds to the RBC and thereby it neutralizes it. So that is another function. Unnecessary vasodilation is prevented by them. Got it? Either I don't think this point you may not get in all books. Okay. So we understood. And viscosity, very important. If in case of polycythemia, viscosity increased or decreased? Decreased. In case of polycythemia, more cells means it is very simple. More thick. More cells, more plasma protein means more thick. More thick means more viscosity, heavier than water. And this is more viscosity is a resistance to blood flow. Okay, when there is resistance, the pressure is on heart. So there is, if it is too much, as in case of polycythemia vera, because of polycythemia, there is a problem, there may be a problem in the heart. Not only this, in the brain, because blood flow, if it is reduced blood flow, when blood becomes thicker, flow will be reduced. So the body may undergo shock, means reduced blood supply to the organs. So in that way, RBC contributes 50% of the viscosity, 50%, other cells plus plasma proteins, they exert the remaining part of the viscosity. Clear? Okay? Hello? You with scarf. Clear? All the points? Very good. Excellent. Disadvantage of hemoglobin. Why it is present in the RBC? Why not in plasma, like plasma proteins? Who can answer? Plasma, this hemoglobin is a protein. So, albumin, globulin, they are also protein. So, they are present in plasma. But why this fellow is within the RBC? What? Definitely there are reasons. So, what is maybe the reason? Now think. Instead of being in the RBC, it is in plasma. What will happen to the osmotic pressure or viscosity? Plasma viscosity will rise. Okay, this is one thing. And plasma protein, the osmotic pressure, colloidal osmotic depends on the amount of, we have discussed. If plasma, if is hemoglobin is placed in the plasma, then what will contain? What will be there? The osmotic pressure of plasma will be much more. Instead of 25, it will be 100. Now you think exchanges in the capillaries. Capillary hydrostatic pressure, oncotic pressure is now 100. Where is that one is 37. Will there be any filtration? No filtration. No exchange. The patient will die, the person will die. So, this is the reason. Okay, what is the reason? Again, try to make it clear. What is the reason? These are the reasons. Biology question, explanatory question of 3 marks. Loss of hemoglobin. Now the hemoglobin is in plasma. Now the blood will be going through the kidney. Kidney will filter it and they will appear in urine and damage the renal tubule. And they will damage the renal. That will be the another consequence. Should I move? Okay. Erythropoiesis I will discuss in detail. It takes place in the processes production of RBC is called erythropoiesis. What are the different stages? And one thing you remember, in which stage hemoglobin starts to appear? In which stage hemoglobin starts to appear? Okay, it starts to appear in this phase. Polychromatophil erythroblast. Chromato means color. Color starts to appear. Means hemoglobin starts to appear. Okay, that's why it is called polychromatophil or intermediate normoblast. It has got another name, intermediate normo. So in intermediate normoblast phase of erythropoiesis, hemoglobin starts to appear within the maturing or development, during the development of the RBC. Okay, and it takes place in bone marrow. In adult, they are synthesized in bone marrow, and in children, bone marrow of all the bones, and increase in number of RBCs is known as polycythemia, decrease is erythropenia or anemia. Okay, these are the places where it is. These are the, okay. Now the RBC structure. So it is have no nucleus. It has no nucleus. Since there is no nucleus, there will not be mitochondria, no use of endoplasmic reticulum. Okay, and there is no mitochondria, no Golgi bodies, no endoplasmic reticulum, no mitochondria, DNA is absent. And then how the RBCs will get their energy? Through glycolysis. Because we know glycolysis takes place only in cytoplasm. But TCA cycle, this takes place in mitochondria. So they cannot derive their energy from beta oxidant takes place in mitochondria. So they cannot metabolize fatty acid. Okay, they cannot metabolize through glucose or carbohydrate through TCA cycle. Okay, or electron transport chain. These are absent. Mitochondria are absent. Okay, and already discussed variation in shape. RBC may have, this is question. Variation, you have to remember this. There are different types of variation. One is crenation. Just you think, one RBC you kept in hypotonic solution. Now what will happen? From the RBC from hypertonic solution. You placed it in hypertonic solution. Now fluid will be moving from RBC to, so this will be squeezed. So this squeezed form of form of RBC is called crenation, shrinkage, as in hypertonic solution. Clear? Very easy. And then spherocytosis, like this. Okay. And condition is in hypotonic solution, if you place, then the RBC will get spherocytosis. Okay. Sickle cell anemia. In sickle cell anemia, they will get a crescent shape. So this is the appearance of a sickle. So it happens in sickle cell anemia, in pernicious anemia. We will describe later. And poikilocytosis, different. If you study the RBCs, there are different forms of different shape of RBC you will get in such condition, poikilocytosis. Okay, so see, you can see spherocytes, these are the spherocytes, these are the normal cells, these are the squished form, and see, here are the different different size, the poikilocytes, elliptical. So this is the elliptical side shapes. And variation in size, macrocyte, microcyte, normocyte. Okay, and anisocytes, within those different size, RBC of different size you will get. So they are anisocytes. Okay, and rollo formation, very important. Question can ask. Rollo formation. What is the rollo formation? All the RBCs are surfaces negatively charged. How they are charged? Negatively charged. So since they are negatively charged, so the RBCs will be repelling each other. Now, if I place more fibrinogen, what will happen? They will cover, they will attach to the RBC surface membrane and minimize this charge. As a result, they will. What happens? The RBCs try to adhere to one another. Okay. As a result, one RBC on the top of this, another RBC means piles of RBC. These piles of RBCs are called rollo. This mechanism of piling of RBCs, because of these changes, increase in number of amount of fibrinogen, so there will be piling of RBCs, and this piling is called rollo formation. Okay, this is the mechanism, rollo formation. So when they form the rollo form, they get the rollo form, what will happen? They will be settling down. They will be settling down, and ESR, erythrocytic sedimentation rate. So what will happen? When you allow the blood to settle, the RBCs will be settling down. Why? There will be formation of rollo. If amount of plasma protein, fibrin or globulin is more, the erythrocytes will rapidly fill down. Okay, so ESR will be fast. Erythrocyte sedimentation will be faster. So we call it increased ESR. These are discussed. So because of the fibrinogen, the RBCs can maintain their suspension ability. When there is a fibrinogen content high, globulin content high, they will be settling down. Clinical significance of rollo formation. In various, be it acute or in chronic, there will be rollo formation. In inflammatory condition, there will be inflammatory condition. There are different stages inflammation. Inflammation is a process by which we prevent the pathogens. We prevent the pathogen from damaging our body. Okay, this is the inflammation. And there are various inflammatory reactions. One is, first, the area will become red. Then, there will be increase in temperature. Metabolism in these cells will be increased. Then, there will be what? Swelling. Then, what will happen? Because of so there will be pain. Then, there will be fighting. antigen antibody, some cells will die, they will die, some cells will, there will be formation of pus. Now this pus will be destroyed or engulfed by the macrophages. Okay, and thereafter repair starts. So these are the different phases of inflammation. Okay, and these are the conditions in which formation is increased. Fate of RBC, in short, RBCs destruction when they get senile, means older, they can't form, their wall becomes very fragile. So when they start to pass through the narrow capillaries, so they will be broken down. And the capillaries present in spleen and liver are more, their diameter is much less. So they cannot pass through this. So as a result, they will be, since they are fragile, they will be broken down. Okay, so when they are, hemoglobin will be released, and now what happens? This hemoglobin is splitted into iron, globulin, and porphyrin. Globulin is made up of, this is protein made up of amino acids. So they will go to the protein food, either they will be stored or they will be split into various amino acids. Okay, now this iron will be carried by the one carrier is there, protein, apoferritin, and through ferritin they will be going to either bone marrow or to the liver for storage and future use for erythropoiesis. Okay, and the porphyrin part will be degraded into bilirubin, and through this bilirubin will be excreted through bile into our GI tract, and they will, because of the presence of the our stool has a particular color because of these bile pigments. Okay, so they will be excreted, and some part of the will be absorbed, and they will be converted into urobilin, and they will be excreted through urine, giving a particular color. Now, hemoglobin. So, hemoglobin is a chromoprotein. Chromoprotein, it is having a colored pigment. Chromoprotein, it is also metalloprotein since it is having iron. And normal values, we have discussed. These per days of free hemoglobin, we have already discussed. Normal variants, normal SBA, adult hemoglobin. 97% of normal human being are having hemoglobin A. Means their globin, how many chains? 4 chains. There are 2 alpha and 2 beta. Okay, let's see what is their globin constitution in different subjects. Remember, its affinity is for oxygen is much more than this. Okay, abnormal variants are SBS, SBC, SBD, SBE, and SBM. What is hemoglobinopathy? We'll discuss what is thalassemia. You have to know hemoglobin, how hemoglobin is synthesized, how hemoglobin is degraded. Take this point. If I am unable to cover, you have to study this. Take these points. And if you take my PPT, from there you will be knowing everything, every point. Okay? So it is the red pigment, conjugated protein, responsible for red coloration. Deficiency is called anemia. And out of him, this hemoglobin, 4% is heme and 96% is globin. Quantity throughout the body, there is 700 to 900 gram of hemoglobin in our body. And this is the amount of hemoglobin per RBC. Their molecular weight is 68,000. Okay, so these are some important topic you have to know. So this is the structure. What is there? In the structure, there are 4 pyrrole rings. There are 4 pyrrole rings attached by methane bridges. So what are these pyrrole rings? So this is one pyrrole ring. That is another pyrrole ring. Pyrrole, and they are attached with methane bridge. Okay, and these corners are having different groups, side chains. This side chain, we number this. We number these spiral rings. This is number 1, this is number 2, this is number 3, and this is number 4. And the chains are the side chain. I mean, the link is alpha, and this one is beta. This one is gamma, and this one is delta. Okay, they are bridges, delta bridges. How many bridges? 4. By 4 bridges, they attach 4 spiral rings. Okay, and all these are also numbered 1, 2, 3, 4, 5, 6, 7, 8. This is methyl. This is methyl, and this is vinyl group. Here you will get a vinyl group, here you will get a methyl group. Here also you will get a vinyl group, methyl, vinyl, methyl, vinyl. And here you will get also methyl group. And here you will get propionic acid. Propionic acid. And it is also propionic acid. Propionic acid, and this is methyl. Okay, now this nitrogen here, there are nitrogen. All the pyrrole ring is having nitrogen. Okay, all of they are having nitrogen. All the nitrogen is having are linked to one metal, iron, that too in ferrous state. If it is in ferric state, this is abnormal hemoglobin, as it is seen in met hemoglobin. It is the abnormal variant. The fingers, body parts becomes bluish, like cyanosis. So that is by that we can diagnose the symptoms. The peripheral part becomes bluish. Okay, so normally it is ferrous state. Why it becomes ferric state? If by some drugs they have effect on bone marrows. Okay, some strong anti-oxidizing agents, they can affect the bone marrow cells. As a result, there will be defective formation of hemoglobin, where instead of there will be, instead of iron, ferrous iron, there will be ferric iron. Now this is abnormal. They will not be capable of performing the transport function. Okay, so now this is heme. This is what? Heme. This iron can bind with oxygen, one molecule of oxygen. It has 6 valencies, 1, 2, 3, 4, 5. Another valence is there. With this it binds imidazole group of hemoglobin, globin chain. Imidazole group of, there is in the imidazole group, there is one nitrogen, it attaches with that nitrogen of the globin chain. Okay, so what is this? Globin chain. One globin chain. Okay. Now in a hemoglobin molecule, how many heme are there? 4. Okay. So all the 4s are having like this structure. So all can bind one hemoglobin and one globin and one oxygen. Okay. So that's why we call it HB4. Okay. When they are oxygenated fully, HB4O. 4, 8, O2 means O2, O8, HB4, O8. Okay. So, there are globin chains. In alpha chains, there are 141 amino acids. And sequence should be maintained. If sequence is disturbed, suppose what I did, She is the sixth amino acid. I replace her with another, now it is an abnormal globin. And now it is an abnormal hemoglobin. Simply I replaced one amino acid. And that caused the abnormal variant of hemoglobin. That are the causes, the abnormal variant of hemoglobin. So now you got the structure of, okay, I got the idea? Okay. See, this is the heme and how the globin chains remain around it. Only one heme remains exposed. Only one heme, normally one, when an oxygen molecule binds with that one, immediately there is change in the structure, conformation, okay, in the shape of the hemoglobin molecule. As a result, the hidden heme moieties comes out. Now they are available for binding with, means binding of one molecule of oxygen increases the affinity for oxygen. Okay, how? By exposing the remaining heme moieties to the exterior. Okay, so that structure is important. Okay, different terms we will be using. And one is oxyhemoglobin. You know what is oxyhemoglobin? Reduced hemoglobin means deoxygenated hemoglobin, no oxygen in the molecule. Carbaminohemoglobin means globin is attached to the carbon dioxide. Carboxyhemoglobin, it is a dangerous one. That is when hemoglobin is having much more affinity for carbon monoxide and then oxygen. So whenever there is presence of carbon monoxide, who will bind? Oxygen or carbon monoxide? Carbon monoxide will bind. And we call carbon monoxide poisoning, because when carbon monoxide binds, it forms a stable compound and it will not be released from there. So hemoglobin will be losing its function. So there will be poisoning, means no oxygen will not be delivered to the tissues. So that is the problem. Okay, and carboxyhemoglobin. So this is carboxyhemoglobin with carbon monoxide. Methemoglobin, I already showed, iron is in ferric form, and free hemoglobin carried by the free hemoglobins. Remember this: when a cell is hemolyzed, what will happen? The hemoglobin will come out. Now who will carry this hemoglobin? Haptoglobulins. Where they will be taken? They will be taking these to liver or to the spleen, where they will be further degraded. Okay. Types of normal variants. One is hemoglobin A, present in 97% of adults. Hemoglobin A2, how it differs here? In hemoglobin, there are 2 alpha, 2 beta, and here, what is there? 2 alpha chains and 2 delta chains. Instead of beta chains, there are delta chains. And fetal hemoglobin, 2 alpha chains and 2 gamma chains. Okay, you have to remember biology questions. These are. And HB bird's hemoglobin, there are, all the 4 chains are gamma chains. All the 4 are gamma chains. And embryonic hemoglobin, there are, when embryonic, during development of embryo, there are different types of hemoglobin you can get. Hello, you are sleeping. 2 zeta chains, 2 epsilon chains. In Gowers hemoglobin, 2 alpha chains and 2 epsilon chains. And now it is abnormal hemoglobin. Number 1 is sickle cell. In sickle cell anemia, you will get this SBS. What is there? 6th. 1, 2, 3, 4, 5, 6. This is glutamic acid. If it is replaced by another amino acid, this will be amino acid. So in this case, glutamic acid is replaced by valine. So simply, it made the hemoglobin abnormal. And what happens? The RBC takes the shape of sickle. And in the previous diagram, they will obstruct the they will be elongated, take the sickle shape, and they will obstruct the capillaries. What will happen? They will be hemolyzed. The fate of these cells are hemolysis. Okay. There is hemoglobin C. Here, lysine replaces glutamic acid. Here, valine replaces. Here, lysine replaces glutamic acid as sixth position. Okay, only this is the difference. Hemoglobin C and hemoglobin D. Here, sixth position of beta chain. Okay, what happens? Glutamine, not glutamic acid. Another amino acid, glutamine, it replaces glutamic acid. Okay, so as a result, what is formed? Glutamine. These are seen only in Sardajis, in Punjabi people. Okay, more common. In Los Angeles, people living in Los Angeles, they are also having this type of hemoglobin. Hemoglobin E replaces lysine at 26th position. Replaces lysine. Glutamic acid, actually lysine is there, so it is replaced by glutamic acid in hemoglobin. And there are unstable hemoglobins that undergoes denaturation precipitation. That means crystals are formed. These crystals are called Haynes bodies. These crystals, and these are seen in spherocytosis. See, they are obstructing the capillaries, so they are lyzed. Okay, these are the normal, these are the sickle cell. So they can obstruct the capillaries and cause hemolysis. Okay, and see, SBM, M type, and this SB type, see, spherical type, not spherical, elliptical type, here it is SBE and it is the SBC. See, in SBC, these are the crystals, Haynes bodies. Crystals, hemoglobin crystals. Since they are crystallized, they will not be performing any function. Hemoglobinopathy, these are genetic disorders. These are hereditary disorder, hemoglobinopathies. And what type of disorder? In the globin chain, abnormal globin chain. And they will cause, they will be termed as hemoglobinopathy. What is there? Structural abnormality. For example, sickle cell anemia. In thalassemia, what is it? Remember, what happens in thalassemia? Everybody knows that. What happens in thalassemia? Defect in globin chain. Either there is less number of globin chains or no globin chain, either alpha absent or less in quantity. Beta is okay. And in some cases there is no beta globin, no beta chain, but there is or it can be like this, there is less globin chain, beta chains. So either there is absence or less. So it may involve only alpha chain, it may involve only beta chain. Depending on that, we classify thalassemia into two, alpha and beta. In the beta thalassemia, alpha thalassemia. In alpha chain, reduced alpha chain. In beta thalassemia, reduced beta chain. These are pathophysiology, time is coming out. Hemoglobinopathy, thalassemia discussed. Hemoglobin biosynthesis. How hemoglobin biosynthesis, a short pathway I am describing here. So what happens? For two amino acids are required. For one is succinyl CoA, another is glycine. So they join together and form a shape like a pyrrole ring. Not complex structure, take a formal ring. And this is called ALA, delta aminoleuvolonic acid. So by the enzyme, see, what is the cell? Where hemoglobin is synthesized? In RBC. Where RBC? In bone marrow. In which stage? Intermediate normoblast. So it is the intermediate normoblast. Okay. Not even it is now RBC. It is intermediate normoblast in the bone marrow. Now intermediate normoblast, this is the mitochondria, this is the cytoplasm. There is nucleus. Now there is a nucleus. During the erythropoiesis, it will lose gradually, it will lose its nucleus. But in this stage, there is nucleus. Okay, so, and there is mitochondria also. Okay, this is the cytoplasm. In mitochondria, 2 amino acids, succinylcholine will form this alpha amino. It is also ALA. It is also in short called ALA, ALA. Alpha lipolenic acid. Now 2 molecules of they will be coming out of from mitochondrial matrix into the plasma. Here 2 molecules will combine and it will form porphobilinogen, PBG, porphobilinogen. Now 4 porphobilinogen. Now already every PBG has taken the shape of pyrrole. Now 4 pyrrole is now attaching through a linearly attaching. Not even now they are not connected by the methane bridge. Now they are linear in pyrrole, 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 pyrrole. Okay. Now these sort of things are uroporphobilinogen 3. Now from here carbon dioxide will come out and it will form coproporphobilinogen 3. Now this will enter into the mitochondria and it will undergo oxidation. It will form protoporphyrin 9. Then it is again it will be there will be oxidation. Will be protoporphyrin 9. Here iron will be added. This iron is carried by the ferritin. From ferritin iron is freed. This iron enters here and forms this and this combined together forms heme. Now this heme comes out and here it combines with globin and forms hemoglobin. Now where from globin is coming? Globin is coming, as I told, how they are come? Because it is having nucleus, mRNA is produced. See, mRNA is coming to the endoplasmic reticulum and here they will be combining with different amino acids and globin chain will be formed. Now this globin chain, our endoplasmic reticulum are present in RBC in cytoplasm. So here a lot of this thing will be there. Amino acids, with this amino acid globin will be formed. Now heme and globin will be joining together to form hemoglobin. Okay, so you have to learn hemoglobin biosynthesis. Okay. Normal levels of hemoglobin already discussed earlier, and high levels of hemoglobin are seen under these conditions. What are these conditions? High altitude polycythemia vera. In newborn, it is more than you, and when there is excessive sweating, means water loss. So blood is becoming concentrated. So per 100 ml hemoglobin also be. So that is another condition. Hemo concentration, as in pregnant women, they become anemic. Hemoglobin is less. But they make their RBC formation at a faster rate. Even then, their hemoglobin is less. Why? This pregnant woman, just reverse, in this pregnant there is edema, formation of edema, isn't it? More fluid accumulation. If from the blood, water moves to the tissue place, blood will be concentrated, hemoglobin concentration will rise. Isn't it? Okay. So this is, and vomiting, water loss, as a result, high levels will be seen. Diarrhea, water loss from the blood, from the plasma, as a result, high level of hemoglobin will be seen. Congenital heart disease. Congenital heart disease is mixing of blood. Mixing of venous blood, and as a result, tissue will feel hypoxia. As a result, what will happen? Hypoxia stimulates erythropoietin, erythropoietin will increase the RBC production. As a result, there will be increase in hemoglobin content. Emphysema. In emphysema, what happens? The patient, he can inhale normally. Problem is, when he is exhaling, the airways become constricted. So he cannot properly breathe. Lot of sound is made. Lot of sound is made. Have you seen the emphysematous patient, asthma patient? So what is there? Always there noise in the chest, no? So why obstruction in the airways? As a result, tissue perfusion, more oxygen is not properly diffused. Disturbance in oxygenation of blood. As a result, tissue will be hypoxic. So hypoxia stimulates erythropoietin secretion. Erythropoietin causes more erythropoiesis. So as a result, hemoglobin concentration rises. So every point you have to explain physiologically. And low levels, these are the conditions in which low levels are seen. Fate. When an RBC becomes agile or senile, it is broken down, as I said. What happens? Hemoglobin is released. Now, this hemoglobin is captured by macrophage. They split it into heme and globin. From globulin, we get amino acids. Heme is split into iron and protoporphyrin. And iron is transferred to transferrin, then again to erythroblast used for hemoglobin synthesis. Okay, and the protoporphyrin undergo in the liver, it is converted into bilirubin. Now bilirubin is conjugated in the liver and form glucuronides, conjugated bilirubin. These are secreted in stool through bile, through starcobilin, through feces they are liberated. Some will be absorbed in the blood and they will be secreted through kidney as urobilin, through urine as urine. Okay, so this is the catabolism, fate of hemoglobin and catabolism of hemoglobin. In detail, we will be studying it because we will be studying jaundice. Okay, what happens in bilirubin? How this bilirubin formed? What are the steps we will be discussing? You have to have 5-6 months. So you have to know it in detail. Thank you.